Well, thank you very much for the introduction, and thank you, organizing committee, for inviting me to these meetings. They're always wonderful and fascinating. Are we starting? Are we? There we go. Clive invited me to talk about evolving technologies, and when I think of evolving technologies, I think of Star Wars. And so you will please excuse my tongue-in-cheek first slide here. But, you know, we think of Star Wars and innovative technology, but, you know, I'm getting old, and I have to tell you, I get up in the morning, I microwave my water for tea, I turn on my flat-screen TV, and the satellite TV, I watch the news. I check my email on my cell phone, and I use my nav system on my car to avoid the traffic jams. When I was a kid, none of that existed. It was science fiction. It was Star Wars. So, you know, here we are living Star Wars, right? So the question is, we're living Star Wars. What's next to come? So, let's see if I can do this. Oops, wrong way. We've been there. Somehow we're not advancing here. So much for advanced Star Wars. You can just go to the second slide if you can get there. There we go. Okay. Hoo-ah. So we're talking about the explosion of technology, and driven from the South. I thought, well, what does Clive want me to think about, driven from the South? And I took that as technology that's driven by need rather than technology for technology's sake. So that's the context I want to talk about things today. You know, we're still not advancing here with me. So I guess you... There we go. It's out. Okay, let's try that then. Okay, so I started doing flow cytometry back in the mid-1970s. And back then, the first generation of flow cytometers had just come out, and you know, I have the list up here. And I cut my teeth on the Coulter TPS-2, which is essentially the instrument I'm showing you there. And at the time, we thought we were Star Wars, okay? We had lasers and, you know, all this stuff, and we look back on it now, and it's so primitive. You know, it was cumbersome. It was so expensive that only a few big medical centers around the world really had these. The technology was limited. We had 2 parameters we could work with. And if you used forward scatter as one parameter, you could only do 1 fluorescent parameter. And to get our data, we literally took Polaroid cameras, which now nobody has because they're antiquated, right? You know, and they were new then. We took our Polaroid cameras and we took a picture of the oscilloscope. That was our data, right? So that's how we started in flow cytometry. So the flow cytometry of the day, particularly clinical flow cytometry, was driven by technology. We had a new toy. What can we do with this new toy? And basically, in the late 70s, early 80s, all we could figure out how to do clinically was DNA cell cycle analysis. That's the only thing that was really amenable to 1 and 2 parameter flow cytometry. Now, my dissertation, which fortunately I got, we tried to automate the pap smear using these parameters. We didn't have monoclonal antibodies. We had 1 and 2 parameters to work with. We got some positive results, and we learned a lot, but we certainly didn't automate the pap smear. By 1981, people had been playing with flow for about 10 years, and Ole Larum wrote this review article on a review of the state of clinical flow cytometry. And you can see that in the introduction, The second line of his introduction, he said the major application of this is DNA cell cycle analysis. So today, hardly anybody in the clinical lab does DNA cell cycle analysis. There are a myriad of other applications, but this has gone by the highway. So what was the tipping point in clinical flow cytometry? What pushed us beyond just saying, well, DNA analysis is good, what else can we do? Well, I submit to you that there was the perfect storm. We had the technology, and the technology was evolving. You know, the flow cytometers were getting better and better. Monoclonal antibodies became available, and we got more and more and more. What was the clinical need? Well, the clinical need, to me, that was the tipping point, was the AIDS and HIV pandemic. We had an immediate need to have technology that we could do things like a CD4 count, do it reliably, do it cheaply. So at that point, to me, we transitioned from flow cytometry in the clinic becoming technology-driven to it becoming application-driven. And by application-driven, yeah, I mean changes occurred. You know, we had standardized nomenclature for the monoclonal antibodies. Before then, every manufacturer had their own terminology. We had smaller, easier to use instruments that were designed just for clinical use. You didn't have to sort cells on them, so they were just analyzers. We no longer had our Polaroid cameras out. We actually could record our data, and eventually we could record list mode data that we could replay. And of course, there's the standardizing of the SOPs, so everybody was doing the same thing, getting the same result, and proficiency testing. So to me, HIV had a significant impact on the practice of clinical flow cytometry. It really changed dramatically clinical flow cytometry. And I think there's no doubt flow cytometry tremendously impacted HIV research, okay? So that's good. I gave you a history lesson. You know, nobody cares. What have you done for me recently, right? So that's human nature. So in this talk, what I thought I would do is present 3 problems and 3 recent innovations that have attempted to that. One of the solutions is fully mature, one is just about mature, and one should be on its way in the next year, I think. So what are these? So problem. Shipping antibodies that need refrigeration and a cold chain. So the innovation is to develop monoclonal antibodies and reagents that don't need a cold chain. Okay, number 1. Number 2, virus latency and examining the heterogeneity of immune responses on a single cell basis. So what's the solution? To create new instruments, new approaches, new assays that have increased sensitivity to detect viral proteins or immune proteins at the single cell level. And then third, what's the problem? Patient compliance. And you can see these are vastly different problems. Patient compliance for monitoring appointments. I'm sure all of your patients show up for everything. At the NIH, there's a problem. If patients are just being monitored and they know they're not getting treatment, they tend to find better things to do. You know, Dunkin' Donuts is having a sale that day. You know, they don't show up. So, you know, what can you do about this? Well, there are devices that are being developed that actually will allow patients to draw their own blood. Okay? So, potential solutions. So let's start with the monoclonal antibodies. So I'm sure I'm preaching to the choir here that, you know, sometimes it's tough to keep reagents refrigerated in transit if there's a long transit time, to keep them stored in the cold. And I have to tell you, even at NIH, my refrigerator breaks, okay? It does happen. Our power goes out. It does happen. We don't like to admit it. So there's a cold chain problem almost everywhere. So there have been 2 innovative solutions, well, multiple innovative solutions, but I'm going to talk about 2 from 2 of our sponsors, actually. Beckman Coulter came up with Duraclones, and these are fluorochrome conjugated monoclonal antibodies that are actually dried onto the side of tubes. It sounds easy, but technically there were challenges to this that took them a while to overcome. And then the other one is from another sponsor, Becton Dickinson. These are lyophilized antibodies. Generally, they lyophilize these in microtiter plates, okay? And these do not require a cold chain. So here we have liquid versus duraclone testing. The liquid's on top, the duraclone's on bottom. On the left, you see CD3 versus CD20, fairly distinct subsets. And you can see that you get good delineation of your T and B cells. On the right is a more difficult phenotype, which is IgD versus CD27 on B cells to detect naive and memory B cells. And you can see that you get virtually identical results with the duraclone. Again, and this is shamelessly swiped from the Beckman-Coulter white paper. Here we have the stability of the duraclones at 40 degrees. Everybody would agree 40 degrees centigrade is rather warm. They stored antibodies for 98 days and compared day 0 to day 98. You can see two different colors, blue and red, and got virtually identical results in a variety of fluorochromes. So regardless of the fluorochrome, the product is very stable over time. For Becton Dickinson, the lyoplates, you can say that they have very comparable data as well. Going from the light scatter on the left, the top is the liquid, the bottom is the lyoplates. The light scatter is virtually identical, CD3 and 20 is identical, 4 and 8 is virtually identical, and your more well-defined subsets are virtually identical. So you get almost the exact results with either of these stabilized reagents not requiring cold chain that you get with your liquid reagents. And again, this was in my lab where we had a panel made up, and that's the other thing, you can make panels out of these things. So this is an 8-color panel, well, 2 8-color panels, one for T cells, one for B cells, that we tested. So this is for deeper immune phenotyping. And again, just you don't have to read the labels. And if you can read the labels, your eyes are much better than mine. I can't read them from here. But you can just see the patterns of the staining is virtually identical for the liquid and the lyoplates. So these are essentially equivalent to the liquids. There's no cold chain requirement, which was the problem, right? So the problem's been addressed. Good shelf life to both of these. They can be pre-mixed as panels. So that eliminates another potential problem in the lab pipetting errors. Manual pipetting errors in the clinical lab is a major source of error. So it can be eliminated by having, pre-made, dried, or lyophilized panels made up. And if you have multiple clinics or multiple labs that you want to get the same results from, you get a higher rate of precision about this. And just in context, we used these reagents in a broader study for the Federation of Clinical Immunology Societies that we published a model for this in Nature Immunology a few years ago. So very good reagents. And I would submit to you that there was an identified problem and an identified innovation in response to that problem, okay? So that's number one. What about number two? Number two is a little bit more difficult. Having ultrasensitive assays for virus, say P24, and for immune responses. So there are a number of new innovations coming down the pipeline in this regard. The one I'm going to talk about is the Simoa from a company, Quantarex. Simoa is single molecule analysis. Now, in reality, don't tell the company I said this. I don't think it gets quite down to single molecule, but it is ultra sensitive. The company has a number of assays that they have on their reagent list, one of which is the HIV P24 antigen detection. They also have a number of cytokine detection kits, such as interferon, IL-17, TNF-alpha. And you can also make a home brew where you can get your own capture and reporter antibodies as well as a recombinant protein to create a standard curve and make your own reagents. And we've done that and had very good success looking at CD146 as a marker for inflammation. Even though this is theoretically capable of multiplexing, it really works best as a single-plex assay due to the high sensitivity of the assay. Now, this assay, if you know ELISA, let's start there. ELISA, everybody knows. That's been around forever. The next generation really was Luminex, the bead array assays, right? It's an ELISA run on beads that you can put through a flow cytometer, and the readout is fluorescence rather than enzymatic. The Simoa is the next step in the Luminex type assays. It's bead-based, but now instead of Luminex size beads, you're using nano beads. And instead of putting these in a flow cytometer or a Luminex instrument, you're putting these nano beads in nano wells, and the nano wells have 50 femtoliter volumes. And because you have such small wells, what you can do is you have blue ray discs that have 24 arrays on them, and within each array, you have 216,000 nanowells. And the basis of all this is you're putting one nano bead in one nano well, and you're getting the readout from a single bead. And that gives you the sensitivity of this. I'd show you a picture of the Simoa up there. We call it the photo booth in our lab. It's about 5 feet high and about 5 feet wide. And, it looks like one of those things at the arcade, the carnival. You go with your girlfriend and have your picture taken. Several people have tried and haven't gotten any pictures out. But this is full, the upper part is just full of robotics. It does all the pipetting, the washing, the imaging. It is all done in the upstairs. The downstairs is basically the sheath, the waste, and all the fluidics that you're going to need. So essentially, with the older technologies, you have basically 100 microliter reaction volume. You have diffusion kinetics going on with your analytes, and you have analog readouts. When you get to the Simoa, you have femtoliter size reactions, so that's 2 billion-fold less reaction size that you're dealing with. You don't have the diffusion, and you have digital readouts. And so it's a whole new ballgame at this point. I'm not going to go into this in any detail, but basically the nuts and bolts of this is it's a fully automated system. You take your samples, you put them in a microtiter plate, and that goes in one drawer along with banks of pipettes. In the other drawer, you put in all your liquid reagents, your antibodies, your enzyme, your fluorescent substrate. That goes in the other drawer. You program the computer, then it will pipette all your reagents, it will wash them, it will add the enzymes, it will add the substrate, it'll do all the washing, it'll put it on the Simoa Blu-ray disc, and then it'll image it. And you don't have to do anything except program the computer ahead of time. You just walk away. And in about 3 hours, you come back and you have your data ready to go. So this is a way of looking at P24 in ways other than like a high sensitivity ELISA. It doesn't require enzyme amplification like the molecular methods do. And the company, when they market this kit, they claim a lower limit of sensitivity of 25 femtograms per ml. But a recent paper that just came out by Wu, I think it was this month actually, they made a couple very minor modifications and now claim a lower level of detection of 5 femtograms per ml. So this is a highly sensitive assay. This is a Journal of Virology paper that came out, I believe, in January of this year. And they compared a traditional ELISA, high-sensitive ELISA, to the Simoa assay. And you can see the black is the ELISA, and the red is the Simoa assay. And you get 1,000-fold increased sensitivity with the Simoa assay. This came out a couple of years ago, and this, I believe the principles were from the company, so take this for what you think it's worth. That was my disclaimer on this. This compares branch DNA results to the P24, and their R-square values are 0.99, so there's a very tight correlation here. And again, now, this is going back to the Journal of Virology paper. They were interested, obviously, in detecting the virus reservoirs and seeing how few cells they could detect that might harbor HIV. And so what they did is they had a GFP HIV particle that they transfected into cells. They sorted these into microtiter plates at either 1 cell per well, 10, or 100 cells per well, and they put these in a background of non-infected CD4 cells. And then they culture these for 18 hours, and they then lysed the cells, and they also looked at the culture supernatants. And on panel C up there, on the left side, are the lysed cells, and on the right are the culture supernatants. And you can see that, do we have a pointer here? We do have a pointer. Okay, here's 1, 10, and 100, 1, 10, 100. You can see that directly and proportional to the number of cells is the amount of P24, and here is your control. And same thing with the culture supernatant, and if you jump over here, this shows the intracellular P24 from the lysate to the culture supernatant. You can see there's a direct correlation there. And we skip down here, looking at the RNA in the culture supernatant versus the P24 in the supernatant. And again, there's a good correlation here. And this one, to me, is probably the least meaningful of all, but it's the GFP mean fluorescence versus the intracellular P24. And there's still a pretty reasonable correlation there. So this assay seems to work pretty well. So certainly this is an alternative way of looking for virus reservoirs in these patients without worrying about inactive proviral genomes that may be around. There may be other implications of the viral protein expression in regards to inflammation and certain kill strategies, where this might be a useful alternative and a rapid alternative compared to the methods which are now being used for this. And again, I refer you to this paper. This technology is very new. As a matter of fact, I think prior to this year, the only publications that had come out were really from the company. And so this year, there have been 4 publications by independent investigators, and I have them listed here. If I talk long enough, you might have a chance to copy down these things. But basically, they're starting to use this technology to look at virus reservoirs in patients as an alternative to the molecular methods. And we're still very early on, I think, in determining whether or not this can be done. Now, my lab is not an HIV lab. My lab is more of an autoimmunity immunology lab. This technology is not just for P24. My lab is more interested in using this in the immunology field, and our question is what I call precision immunology, looking at the immune responses of single cells. Can we do that? And the answer basically is yes, that we took fresh peripheral blood, isolated the PBMCs, identified cytokine-producing cells using surface bispecific antibodies, sorted these, into microtiter plates at either 1 cell per well, 2, 5, or 10. These wells had a lysis buffer. We put the lysates in the Samoa and said, can we detect cytokine production in an individual cell? And the answer very quickly is yes. This is 1, 2, 5, 10. And these are our interferon negative cells by surface marker. And we found an average of 34 femtograms per cell of interferon gamma. And this is, right here is the number, 3.4 times 10 to the negative 17th grams. That's not a lot, but we can detect it, and we can detect it reliably. It's not a one-trick pony. We can also do this with TNF, and we can also do this with IL-17. So now you're going to be able to really look at immunology in a super precise way. So that was kind of a quick overview of that. So you can look at P24, you can look at all sorts of immune responses, and I will say you can look at other analytes that I haven't even thought of yet and may not have even been discovered yet. You know, anything you can squeeze out of a cell, you can look at probably a femtomolar or maybe even attomolar level. So exquisite sensitivity. So I would say, you know, we'll put a check mark on this second group here that, you know, there are attempts to actually look at different analytes for virus latency and immune responses at a level that has not been possible before. So what about patient compliance for monitoring appointments? Well, there's a technology that's being developed which I personally think is really cool. And so, you know, what if you could get patients to draw their own blood, right? I mean, think about it, you know. Not only would that be convenient, but you wouldn't have to hire a phlebotomist anymore. You'd save a little money. There's a device called the Hemalink that is being developed by a company called Tasso. And while you may get some people to stick a needle in their vein with a vacutainer on it, you're probably not going to get a lot of people to do that. So what they came up with was, you know, let's go for something that uses lancet-based blood drawing. You hide the lancets so people don't look at these scary sharp things coming at them. They really don't have to do anything. They just push a button and it takes 2 minutes to get the blood. That sounds pretty simple. So the device looks like that, and I don't have a functioning one, but I've got just like, that's what it would look like. And the idea is you just come over here, you put it on your arm, you press the button, and you go make your cup of coffee, right? And by the time your coffee's made, your blood's drawn. So that's the idea. So you like that. So here's today. You drive to the clinic, you take the bus, you walk to the clinic, you wait for the phlebotomist to get around, you draw your blood. They want to move to this scenario where you can stay at home, you can put it on yourself, and then you can either put the little capillary tube or Eppendorf tube, I'm sorry, in the mail, or you can have a courier just run around, you know, if it's local and pick up these things. So you don't get a lot of blood. Right now, they're getting about between 150 and 200 microliters of blood. They're working on ways to up the amount of blood that you get. And one of the things that they're doing that works well in North America is you just, you know, put some a heating pad on your arm for about 30 seconds, and that increases the blood flow to the surface. It's probably not a problem down here, I imagine. So, you know, your volumes of blood might be a little higher, but there's still a lot that you can do with certainly 300 or 400 microliters of blood. This is very much a work in progress. They're working on stabilizing reagents and doing antigen-specific stimulation tubes, if you're interested in that. A major thing is comparing values between capillary blood and venous blood. That could be apples and oranges, so that could be a major sticking point, that if there are major differences, it's probably not going to work out well. And of course, increasing the amount of blood would always be good. Although I don't think it's crucial to get to 1 ml, it would definitely be worthwhile. So I'd submit to you that I'm going to put a check mark by this, that certainly people are working on it. It might not be the ultimate solution, but people are thinking about it and working on it. So I think we have undergone this dramatic change since the 80s of being technology-driven to being need-driven for a lot of what we do. And I think that's going to continue with the resource limitations and the growing need and awareness of these needs. And so, you know, I guess I would go back to Star Wars, you know, dare to dream on this. You don't have to have a solution. You just have to have the problem. And people have talked about the HIV stigma. Don't have technology stigma, right? If you have a problem, you know, talk about it, because the odds are other people have the same problem. And the only way your needs are going to be addressed is if the engineers and the companies, people hear about that, because they want to address these needs and they're willing to. And I think this kind of shows that they're doing it. They're actually working on it. So I would encourage people to you know, talk about the need. Don't talk about the solution, because, you know, there are lots of great engineers that will figure out the solution. Just talk about what you need. So I'd like to thank some of the people in my lab that have contributed to some of the data I showed, and to Holden Maker at Stanford, who is my partner in crime on the lyoplate and Dur clone studies, and Roger Patak at Southern Research, who does a lot of the P24 testing with Samoa and otherwise, who helped me out on that part of the talk. So thank you.
So thank you very much, Phil, for your very enthusiastic and promising story. I think you not only give us the feeling that we are ready for the next decade, but also the data. I'd like to open the floor for the audience to ask some questions for Phil. Are there any questions? Yes, there's the first question.
Hi, Phil. Sten Vermont from Yale School of Public Health. You know, in the old days, we have 96 wells and we have automation, and you can easily see how contamination can be avoided. I can't get my arms around over 200,000 cells, I guess, for lack of a better term, on a Blu-ray disc. How can we be that precise to avoid cross-contamination across the little wells in that incredible matrix? I'm not a lab guy, so this is coming from an epidemiologist. I don't have good instincts for how the engineering functions.
Right, and I will do my best to answer that with a caveat. I'm not an engineer either. I love the technology, but I'm not an engineer. But basically, what this does is there's a very small volume to these nano wells, and of course the beads will take up a certain proportion of that volume, just one bead. And so the sample fluid with the beads is layered on these arrays. And then there's a layer of oil put on top that will prevent any movement back and forth. And then the whole array is imaged by CCD cameras. So you're actually looking at the bright spots in each well, and you can identify the wells that have none and the wells that have one. And I'm not sure the wells are even big enough to accommodate 2 of the nano beads. If they are, and I don't think they are, but if they are, you can identify those with your CCD camera imaging, which is all part of the electronics. Yeah.
Glad to hear it's not whirring around.
Yeah. Well, it does whir around a little bit, but not that much.
Excellent talk, Philip. The pre-selection process seems to be kind of important when you're looking at like one in 100,000, one in 10,000, et cetera. Can you tell us a little bit more about the pre-selection process before you're putting these things into the Samoa?
The pre-selection process of the cells? Well, now, my experience in my lab has been with the cytokine events, okay? And so what we do with the cytokines, you don't want to stain intracellular cytokine because then you've bound up all the analyte that you want to measure, right? So what we do is we take bispecific surface antibodies. So one arm is CD45, the other arm is a capture for interferon gamma, right? So we take our cells, we throw them into culture, and then the next step is we stain these with CD3, CD8, and our bispecific antibody that's been in culture. And so we're now capturing the cytokine-producing cells. We can identify them based on the surface staining without going intracellularly, okay? Yes, they have to be secreting at least some amount. And then, on the basis of that, we set a gate on our flow cytometer and sort them at a given number into the well. So they are pre-selected. The opposite side of that are our controls are from the same patient, with the same criteria, except they're a tight gate around the surface cytokine negative cells. And we have shown actually by preliminary experiments staining surface and intracellular at the same time that there's a reasonable correlation of the two. Yeah. Because you're absolutely right. We had to have a positive control here to go after them. Yeah. Basically, in these studies, they've done things like transfect with HIV, and then put a background of negative cells. What you would do otherwise is, you would take cells and you would use a latency reversing agent, and then just take whatever number of cells you're going to use for your assay, 100,000, a million, 10,000, whatever it is, slice the whole thing and measure it, with the idea you have kind of a standard curve that you've generated to see, if you're at the 1 level, the 10 level, the 100 level, like I showed, right? Well, the P24 would be presumably coming from a single cell if it's a certain level of P24. So in other words, you can generate the standard curve by artificially making, okay? So you know the level of P. Well, it doesn't matter how many negative cells you have in the prep. It's you have a positive cell, and how much P24 is there per positive cell is the question, right? Right. If you know that, then it becomes more straightforward. You buying that? Remember, you talk after me. I'm going to ask you terribly hard questions, Mom. You're a dead man. I have a long memory.
Okay, one more question. One more question.
Make it gentle.
It is. Bharat Parekh from CDC. My question was about Hemolink.
Yes.
Do you know the cost of the device, or is it? This could be very useful for many settings.
I don't think as far as I know, they haven't set a price for it yet. My impression is it's not going to be extremely expensive. I don't even think it's going to be very expensive. It's a very simple device. It's basically a suction, kind of device with one button on there.
It's relative, expensive in US versus outside.
Right.
I'm talking about Africa.
I just have no idea what the price is going to be on that. Sorry.
Okay, thank you, Dr. Phil McCoy, for your presentation and the audience for the discussion.